From Microbe TV, this is Immune, episode number 63, recorded on December 15th, 2022. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi again. Welcome.
Where the snows are starting to fall, right?
They are. It's rain right now, but it's about 32, so it's just creating a nice slick ice cover of everything, and then it will turn to snow, and then we'll be okay.
Here it's raining, so we don't have to worry. From Cleveland, Ohio, Steph Langle.
Hi there. Great to be here. We have very similar forecasts for our weather. It's going to be raining, then ice, then snow.
And from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is just gray right now, and I think we're not going to get nearly as much snow as some of the other folks.
Maybe next year you'll get more.
That's okay. I don't really mind. I think I've had a lifetime supply growing up, so.
Yeah. I feel the same way. I don't. It is pretty though when it's fallen, right? Someone just sent me an email from the UK, and they, these stickers. People are sending me pictures of where they put the Microbe TV stickers.
Oh, that's cool.
It's cute.
That's a great idea.
It was a picture out their window, and it was London, and the buildings were covered with snow.
Oh wow!
Oh, that's nice.
And they were complaining it's cold, but it was really pretty because snow can be so pretty.
It is pretty here too. We've had a couple of snows already this year, and it was a little heavier wet, so everything was coated in white. It looked like winter wonderland. Nice. The coolest though is when we get those big giant flakes coming down, and it looks like you're in a snow globe.
Mm-hmm.
So this is our last Immune for 2022.
Wow, look at us. How many we've done at least 12.
I think we did 12.
I think we did more, maybe just 12.
But the year goes by quickly. And so we thought for this last episode, we would do a little mixture of things. We'd have each of us pick a couple of papers and chat briefly about them.
We might have to be brief. I was looking at the emails. We do have a lot of emails if we want to get to them, plus our papers.
And I have another pod at 2, so we have to finish by then. Okay. So we have 2 hours. That's good. And then emails. Yeah, I collected most of the emails. And many of them are, could you discuss this paper sort of thing, which, we're not going to do now, but we could talk about future. Yeah, we'll do this in the future sort of thing. So let's start with some papers. And
you want to do a round robin? We all kind of.
Yeah, that's a good idea. We could each do one. Sure. So here the order would be, I want to go last, okay? So it would be Cindy, Steph, Brianne, and then me. I'm going to move mine to the end because I'm just here in spirit as an immunologist.
No. We give you an honorary card.
An honorary degree. All right. So Cindy, you start us off.
All right. So the first paper I picked, I'm going to call CAR-T for SLE. So cool. You might remember that we've talked about CARs a couple of times. So on Immune 4, we talked about CAR-T therapy. And then I also did an update on CARs in Immune 43. And there we talked about using other cell types other than CARs, but in both we kind of touched a little bit on, other than using these chimeric antigen receptors or CARs for cancer, what else might one be able to use these for? So a paper came out in September of this year in Nature Medicine, and it's entitled Anti-CD19 CAR-T Cell Therapy for Refractory Systemic Lupus Erythematosus. And it's by Mackinson and colleagues. And just as a quick background, so SLE or systemic lupus erythematosus is an autoimmune disease with about 0.1% prevalence, and it's much more common in women. And the disease is manifested by the production of autoantibodies against various different antigens, which are mostly nucleic acids, but there are other things as well. And I'm interested in this because nucleic acids are recognized by TLRs by the immune system. And it turns out that these immune complexes will trigger a feed-forward loop where they cause antibodies through the stimulation of TLRs to produce more autoantibodies. And so some of the things that people have thought about treatment here are blocking that feed-forward loop, right? So that's something I've been working on. How do we regulate TLRs to understand how maybe we could interrupt their signaling and then like block this stimulation of B cells to produce these autoantibodies? But they're not the only cells involved. The DCs and other immune cells can take up these immune complexes. But regardless, there's really no cure at all for this disease. Treatments center on immunosuppression and symptom management. So there's not much you can do for these people. But knowing that B cells, which make the antibodies, have markers which could be targeted, this has been tried in various different ways. So one of the ways is a monoclonal antibody therapy, which is called rituximab, and that's an anti-CD20, which will bind to mature B cells and cause mature B cells to be removed out of the body. But some patients are refractory to this. So people are still thinking about this and what might you do? Now, CARs are chimeric antigen receptors where you take a part of an antibody targeting an antigen of interest and you fuse it to a T cell receptor signaling domain. Then you can put it into killer T cells, put the killer T cells into the body, and they should go seek out whatever cells have that antigen on them and destroy them. For cancer, they've been doing this. Works extremely well for lymphomas, B cell cancers, because you can take CD19, which is on most B cells, and you can give them anti-CD19 CAR, and the CAR T cells will go and they'll just eliminate all of the lymphoma B cells. They wipe out all the regular B cells too. So how might one think about doing this is, well, if you've got autoantibody producing B cells, why don't we just wipe out all the B cells? Then maybe the disease goes away. So in this paper, they treat 5 patients. So this is actual human study, 4 women and 1 man, because this is more prevalent in women, right? So we don't have an exact balance. And they use CAR T therapy. And the CAR is an anti-CD19 SCFE, or part of an antibody fused to the T cell signaling domains. They take the patient's T cells out. They transduce them with the CAR. They put them back in. And they saw rapid depletion of B cells within 3 days, which is expected. The T cells expanded in vivo, which is nice too. Their white blood cells dipped a little bit, but they returned back to normal by about day 15. But the B cells remained gone for 30 days at least. So there was, as expected, a dramatic reduction in autoantibodies. But the real important question is, what did it do to the disease? So disease is measured by this thing called the Systemic Lupus Erythematosus Disease Activity Index, or SLEDAI, or SLEDAI, or however they say it. And what happened is they achieve complete remission in all 5 patients for the duration of the study. Wow. With a follow-up of still maintaining a disease index of 0 for at least 12 months after the infusion. So this is pretty amazing. At that time, there weren't any CAR T cells left, but the patient's B cells started to reappear. So they wiped out all of the B cells, but then there were new B cells that were made, and they were naive. They were not class switched. They were not autoreactive, so the autoantibodies stayed away. And the last important thing that they measured was this CAR T therapy comes along with it the cytokine release syndrome, which could make it really difficult for patients to tolerate this. But for whatever reason, maybe they were already highly inflammatory or they were used to feeling bad, but it was really well tolerated. So they did not have a lot of cytokine release syndrome in these patients, but they had complete remissions for at least 12 months, which is pretty amazing. So I thought that was a really cool paper.
That's very interesting. And do we understand why women are more prone to this disease? Is it hormone based?
There is a lot of things. There is definitely a lot of studies on hormone, and hormones can play a role. You probably are aware that a lot of people are doing studies now in male and female, right? Because there are differences, and females tend to be stronger responders, for example, to vaccines, and also have more immunopathology and are more prone to autoimmune disease. There's probably some genetic things as well, but there's definitely hormones connected to that, and sometimes there's some X-linked genes that amplify immune response as well.
Very cool.
So this is very exciting. I just wonder if, after it wears off, do you have to have another infusion? And is that, I guess that's okay because that's what you do for cancers, right?
I don't know how much you can reuse these because you're probably going to generate some immune response to this completely novel antigen that you've now introduced into the immune system.
Yeah.
So I don't know about repeated treatment, but
I guess the question is, how completely do you get rid of the autoreactive B cell in the first place?
Yeah.
If you really get rid of all of the autoreactive B cells, then you're, assuming that you're going to repopulate the immune system with naive B cells. And what's the probability that you're going to get the same cell with the same autoreactive specificity, avoiding tolerance mechanisms again?
Right. And I'm sure that could happen, especially if there's a genetic predisposition. Right. I guess if you didn't develop lupus until you were in your 20s and then you get this treatment and then you have another 20 years of health, that's probably not a bad thing. I don't know if that's going to be the case or not, but we do know for cancer, there was at least one example of the very first person who was infused with CAR Ts. They still are lymphoma free. So that's a little bit different though, right? Because like you're saying, if you already have these autoantibody producing B cells and you have, more naive ones available, could they then cause problem? It's possible it can come back, sure.
Very exciting. I guess they have to do some bigger studies before it would be more broadly used, right?
Yeah, definitely.
Cool. All right, Steph, you're next.
Okay, so my first paper that I want to discuss is called Maternal Gut Microbiome Induced IgG Regulates Neonatal Gut Microbiome and Immunity. And this is building on work in this field of maternal induced effects on the neonatal immune system, and specifically, how do maternal antibodies regulate the gut of the baby, or in this case, they're using mice. And so this is in Science Immunology. The first author is Catherine Sanidad. It's at a Melody Zhang's group at Cornell, but I believe she's at Cornell Medical College.
Yeah.
So what the question that they're asking is, how do breast milk antibodies, and so you'll see a theme with both my papers, but how do breast milk antibodies, how do they manipulate, or how do they kind of coerce the gut bacteria to behave in a certain way that's protective or not? And so what they identified is that antibodies in breast milk that are against bacteria found in the maternal gut were protective against pathogenic bacteria, but also you could immunize the mom or give the babies antibodies against the gut bacteria to see the same effect. And so if we think evolutionarily about why would there be antibodies in breast milk that are targeted against the bacteria in mom's gut? First of all, we have to think about how those antibodies got there in the first place. Well, mom's B cells are constantly circulating during lactation. The mammary gland tells these cells to come here. It's time to, there's a lot of chemokine induced activity. Those cells can set up shop. The antibodies can also come from the blood. But there's this very strong connection between the maternal gut and the maternal mammary gland. And it would make sense that a baby who has antibodies against mom's gut microbes is better set up to establish its own microbiome because it's going to see the same bacteria in its environment. And what I loved about this paper is, again, this isn't, the concept isn't new, but it just further builds on the evidence that this is possible. It's very hard to study this in humans because there is no situation where we can isolate just breast milk effects in humans. We can look at associations, but there's always placental transfer in babies. There's never a case where there's no placental transfer. So it can be very hard in humans to distinguish what are the contributions of these two, either placental IgG or breast milk. And then the mouse, they were able to do that with genetic tools. And you can immunize for this. So let's say there is a gut bacteria in mom that antibodies against it promote a good microbiome ecosystem in the baby. Well, if mom isn't producing many of those for whatever reason, maybe her gut microbiome is dysbiotic, you could immunize against a piece of that bacteria, and they do that to generate antibodies against that bacteria to end up in the baby's gut to promote this symbiosis in the baby. So it's very similar in the way I think about maternal immunization against pathogens. So if I want to immunize mom against rotavirus so that her rotavirus antibodies are higher in milk to prevent a pathogenic disease. It's the same thing, but you're trying to harmonize the microbial ecosystem. It's no longer about just blocking a pathogen. It's about good antibodies that bind bacteria and say, hey, we're going to hang out in the gut longer. I'm going to stabilize you so that as the microbes come along in infant development, we can just, and specifically they talk about dampening IL-17 related inflammation, that would not be a cytokine that you would want increased in a developing gut of an infant.
So I am really interested to know how this influences these mom swaps, right? So when they've been doing all these microbiome studies, they'll nurse pups on a mom that has a dysbiotic microbiome or vice versa. And it seems like it's not just the microbiome, it's going to also be the antibodies that the mom has that's influencing the microbiome, which...
Exactly. And it does kind of blow the mind. One human thing we can look to when we think about the impact of mom's milk over donor milk is NICU infants. So they do better with mom's milk than they do donor milk. And this could be why. It could be that mom has antibodies against her own gut microbes, which in infants with necrotizing enterocolitis, it is the bacteria that are like attacking the infant's gut. So it's really a really interesting way that nature has we're evolved to protect the babies from mom.
And in some ways it to me was a little bit counterintuitive in some parts, because you're absolutely right that having antibodies to the microbes in mom's gut microbiome makes sense, because that you're going to see those same microbes in your environment and you want to protect against them. But part of me also says, well, if they've been potentially healthy in mom, don't we want to keep those and block maybe other types of microbes from colonizing that could be dangerous? And so this isn't what I would have expected necessarily. I would have thought that that mom's microbiome might have been, we might have had a system to more closely replicate mom's microbiome in the baby, because perhaps one that was healthy in mom might be healthy in the baby. And so I think that's really interesting to think about, why did it not work the way I was expecting?
Yeah, and I'm glad you brought that up, because there is this whole concept of how do bacteria behave differently where they're either bound or unbound by antibodies. So we think about antibodies targeting a virus, a bad bacteria, but when you do these, they call it bug seek, that you can pull out the bacteria that are bound to antibodies and sequence them and see what's going on. Mostly they are beneficial bacteria that are bound. And so the way I think about this, kind of like bunch of different ways antibodies can act. They can neutralize bad things. But if you have IgA, for example, that's enchaining these bacteria in the mucus of your gut to create a layer so that when Campylobacter or E. coli or something bad comes, they've created a barrier and it will not penetrate to that. So I think, right, Brianne, what you're saying is a way that could make sense is that antibodies against good bacteria in mom make it to the baby so that it will lock it there. It will say, bacteria, you're good, stay here.
That's really cool. And also I like the word enchaining.
Oh, I do too. That's good. I just wanted to be able to say that out loud. Cool. Well, thanks.
All right, Brianne, you're next.
Sure. The paper that I wanted to talk about is called Long Primed Germinal Centers with Enduring Affinity Maturation and Clonal Migration. Cindy had fun titles for her paper, so I guess this one is sort of about persistence of memory.
So many great words in there.
Yeah. And the first author is Lee. The last author is Shane Crotty. These data were presented at the Society for Leukocyte Biology meeting that we were at earlier, and this was published in Nature in September. I find this study really interesting because it sort of addresses a number of different themes that I've thought about at different times. And I remember when I saw these data presented, I immediately was texting this paper to an awful lot of people. So when I was in graduate school thinking about memory responses, though it was more CD8 memory responses, one of the things that a lot of people were working on had to do with the persistence of antigen and what happens if you change the amount of time that antigen was present. How did that change the memory response? There was a whole kind of arm of papers that were looking at this question of what happened with changing the persistence of antigen and how did that influence memory development. So I've always kind of found that question to be an interesting question. And of course, we've thought about some of these types of things, particularly with germinal centers and how long do germinal centers last and things like that, as we've been thinking about vaccines and when to boost. And as we've seen some of the data on things like hybrid immunity, I keep seeing different types of response, people, coming up with different data on germinal center reactions perhaps lasting longer than I might have expected. And it's something that I've seen in a number of places and always found really intriguing and realized that it was something that we didn't maybe understand as well as we could have. And I think that Cindy might touch on some of this theme of germinal centers later as well. So in this study, they used an HIV envelope protein immunogen in rhesus macaques. And they either used a standard protein immunogen with alum, a standard adjuvant, or they used a new delivery method for the immunogen that allowed that immunogen to be present over a longer period of time. So they had this antigen persisting over a somewhat longer period of time. And it's not, crazy long. It's 12 days. But it's certainly longer than an original protein antigen. And I also think that that's sort of interesting if we think about what it might look like with an actual pathogen, which is probably present longer than a protein immunogen would be. They also gave the macaques boosts a little bit later. But what was really interesting is that the macaques that received the nanoparticles that allowed for longer antigen presence, so basically that gave them that persistent antigen for 12 days, had dramatic changes in their germinal centers. And that was the thing that I remember seeing in the data. Actually, it's pretty much figure 1B and C that I remember seeing presented and sort of having my jaw drop, where the sort of percentage of germinal center B cells was dramatically higher when that immunogen was delivered over 12 days. And so looking at the amount of mutation, looking at the number of proliferation cycles that the B cells had undergone, you could see these germinal centers going out to and persisting for almost 200 days. They mentioned at one point that the B cells were 49-fold above baseline still at 29 weeks. And so they were seeing these incredibly long-lasting germinal centers, which is a period of time that I never would have expected for a germinal center and made me realize I needed to go back and learn. And I think a lot has been learned in the past couple of years about the germinal center, and maybe we all had a lot to go back and learn. But I also really like the fact that I think a 12-day antigen may have some ways that it's similar to an antigen that is seen with a microbe. And this might tell us more about what a germinal center really looks like. And I just found it fascinating.
So you mean like in a natural infection, this is more, and that's why the system has evolved this way, right?
Exactly. Yes.
Yeah. That makes perfect sense. Yep.
We did this paper in our immunology journal club here, and we kind of picked apart the alum thing because it's one dose versus there's a pump that delivers it continuously.
Yeah,
right. It's okay.
Yeah.
I mean, the one dose is how we do all the vaccines now. So looking at doing it a different way and seeing a different outcome is really important. One of the things that we had a conversation about is, they put a pump on these mice and it delivered like a dose every day for a certain amount of time. But how would we do that for humans? You're not going to go to the doctor every day. And so, we're like, there's, materials science people that are doing this all the time. They can cage things that have shells and they deliver things over time.
Yeah, slow
and we also talked about, they're developing these little like nano needle patches.
Right. I was thinking the micro needle patches.
Put the patch on, you take it off the next day, you put another one on, and you just do it for a week. And then, maybe that's a much better way of doing vaccines. Because we just say, you've always done it this way, right? So we always just do one shot in the arm. And then we've been arguing about how much time do you put between the first shot and the second shot. Right. But maybe this is, we shouldn't be doing it that way. We should completely rethink how we're doing it.
Right. But also, how long do you think a sort of primary response, a primary germinal center really lasts? And whatever answer I would have given you before this paper, 191 days would not have been it.
Right. Right.
And so when we think about different viruses that we're trying to immunize against, this is particularly relevant to something like HIV, where you need affinity maturation over a longer period of time to try to develop antibodies that are cross-protective against all the HIV variants that are going to be in your body. Am I
absolutely? Yeah.
Correctly?
So
Right.
In many of the HIV broadly neutralizing antibodies that people have seen, though there is another paper that came out recently, slightly different than this, but that's okay. I thought about talking about that paper too. That you do seem to need a lot of somatic hypermutation and particularly long CDR regions to get that neutralization. And so you'd need many rounds of the germinal center reaction to happen to get that to occur. And so with sort of a complex immunogen, thinking about ways that we could extend the length of time that the germinal center process was happening could be very helpful.
Right.
All right. Very good. So I have 2 of my 3 papers have to do with antibodies and
antibody heavy. Yeah. Round robin. That's good. It's my favorite.
Mine is looking at the FC part of the antibody is important because everyone, when it comes to viral vaccines, seems to focus on neutralizing antibodies, which is the top part of the antibody, which are really easy to measure. It's easy to do a neutralization assay. And I think that biases what we think is the role of neutralization in protection. So I have 2 papers, one with 2 different viruses that address this. And one is a preprint actually, but I thought it was worth doing. It's from a really good group. It is in bioRxiv. Fc gamma receptor dependent antibody effector functions are required for vaccine protection against infection by antigenic variants of SARS-CoV-2. And so this is from the first author is Samantha Mackin, and then we have Galit Alter and Michael Diamond and Ralph Barrick on this paper as well. So basically, the theme here is lovely, right? We're using a vaccine against COVID-19 that is based on the ancestral virus that first emerged, Wuhan 1. But they say, the mRNA vaccines based on the spike of that ancestral virus can still protect against severe disease and death caused through Omicron, right? And they say retain their ability to protect against severe disease and death, suggesting that other aspects of immunity control infection in the lung. So they say, then they say, okay, so maybe it's more than neutralization. Because with Omicron, the antibodies induced by the ancestral vaccine don't neutralize the virus, especially the current variants, yet still there's not a lot of severe disease and death. So they're saying that maybe it's FC functions. And the FC part of the antibody can bind receptors on various cells. It could be natural killer cells. It could be on macrophages leading to uptake. And so they say, it looks like FC might be important, but nobody's really rigorously tested this. So they use experimental approaches to address this. They have wild type and FC gamma receptor knockout mice. So you can see if taking out the receptor for the FC makes any difference in in protection, right? So then they passively transfer immune serum to mice, and then they ask if they're protected from infection. So we're just looking at infection here. And they say this protection is lost when you don't have certain Fc gamma receptors, or if you deplete the mice of alveolar macrophages. And then if you immunize the mice with the mRNA vaccine, the protection conferred by that goes away when you have mice that lack Fc gamma R3, a particular gamma receptor. So, and then again, this is infection of the lung, and they say this suggests, that you need Fc functions of antibodies and macrophages, and that's possibly why in people we still see protection conferred by these ancestral vaccines. And I think it makes a lot of sense because you can say that neutralization correlates with protection. That makes sense because if you neutralize virus, the virus is gone. But in a host, it may be more than that. You have other contributions, and if you don't measure them, you don't know they're there, like the Fc effect. And then if you lose the neutralization and still get protection, it could be as a consequence of the Fc. So I like that people are starting to look at this. Here we are 3 years in, and people are starting to look at this. And we'll see another paper with another virus. And then a third paper looks at the role of T cells in this same issue as well.
Yeah, it's been great to see the appreciation of the Fc component, where for so long it had been Fab biased. And I think you're right. It's easy to measure. I do know now HIV is, I think, unique. So it might be a virus-dependent phenomena. I know when they infuse rhesus macaques with just the, with non-neutralizing HIV-specific antibodies, it does not protect the monkeys against SIV. Mm-hmm. But for viruses like SARS-CoV-2, RSV, it seems to work. And, I think in a seronegative population, would I want just non-neutralizing? No, I want neutralizing. But once we build those layers of immunity, like you said, it's all contributing. So my question is, though, and I'm sure I know people are working on this, but how do you drive Fc functions with immunization? So we know how to drive Fab region, but how do you make Fc binding to a macrophage better or worse with the vaccine?
I mean, my first guess is probably that this is where things like the adjuvant, and how you get your T cell help and how you get your innate immune stimulation become really important.
Aren't there also glycosylation variants that are better at binding as well? Right?
Yeah. There are. I almost picked one of those papers.
Yeah, because if we could harness that, because Vincent, like what you're saying, they clearly are protective. So why not? If we can find a way to, what adjuvant would induce both neutralizing and non, or Fc, associated? And
as you said, this depends on the virus, but clearly SARS-CoV-2 and RSV are important. And then we did a paper on TWIV this year also from Michael Diamond's group on Myarovirus, a mouse virus, but showing the same thing, that neutralization 100% correlates with protection correlates with Fc functions, not with serum neutralization assays. So I think as more people publish this, there'll be more appreciation and more work, which is good. Right.
So there's no concern that if you have a virus picked up by an antibody and binding to an Fc receptor, that it might not gain neutropism?
So now I'm feeling sad that I didn't pick. But that was one of the things. There are some papers that talk about how that is glycosylation site dependent.
Of course, that's what gives you enhanced dengue, right?
Exactly. Yeah.
When it gets into cells that it shouldn't be in because of this interaction. Yeah. So that.
Well, maybe though we could separate out that effect by if the virus infects a macrophage or a monocyte-derived cell, because I think the problem with viruses that infect those cell types, everything comes in very close contact with each other because the virus is replicating in the same cell that's going to be activated by the Fc.
Right.
Yeah.
So, I was thinking about that.
Is it easy to predict what host cells will support virus survival and replication?
No. Yeah. You have to do it experimentally, right?
Yeah.
Impossible to predict. People try, but they fail.
Yeah.
All right, back to you, Cindy.
So my second one I called the dark side of macrophages. Nice. So I saw some of this work published previously or talked about previously, and I just happened to come across this paper that was published in Nature Medicine in November, just like last month, a couple weeks ago. And the idea here, the title of the article is Inhaled Particulates or Particulate Accumulation with Age Impairs Immune Function and Architecture in Human Lung Lymph Nodes. It is by Ural et al., and it is from Donna Farber's lab at Columbia. Donna's lab has been collecting human samples for a long time, and they have a huge repository of various different organs that they're investigating immune cells within these organs. And they had this curious observation that whenever they would collect lymph nodes, some of the lymph nodes were black. So if you've ever opened up a mouse or any other animal and you look at lymph nodes, they're kind of like a tannish, brownish color. And in fact, they look a little bit closer, and it was only the lung lymph nodes that were black, because if they took the mesenteric lymph nodes, so the ones in around the intestine, from the same individual, they were fine. They were not black. So that's weird, right? And they checked for the first thing you would think of would be smoking, right? So if somebody smokes, right? So they accounted for that. So these are all non-smokers. Why would they have black lymph nodes, right? So they are thinking that there's an environmental something going on and the question is, we've known for a long time that the environment can impact the immune system, but exactly the mechanisms of how this works is not clear. But we know that lymph nodes drain the fluids out of various different tissues. So if you have lung draining lymph nodes, they're going to be draining liquid from the lung, and that particulates can be in there. And there's actually specialized macrophages that are in lymph nodes that basically pluck things out of the lymph and bring them into the lymph nodes so that B cells can react to them. And I thought, that's probably going to be those. They're called subcapsular sinus macrophages. They're lining the little conduits, the afferent lymphatics that come in from tissues. So if you breathe in some particulates, they're going to get into the lymph. They're going to drain down these subcapsular sinus macrophages will take them up. Side note, did you guys see a paper a while ago that macrophages maintain tattoos because they eat the dye and they maintain the dye? So that's cool.
Wild.
So there's black macrophages that maintain tattoos.
So the macrophages stay there?
They stay there, and if they die, then another macrophage eats them, and then they keep the dye. So that's how tattoos stay. Anyway, side note, I just thought that was a really cool thing because they're also like dark colored. But they do a whole bunch of things, but the idea here is that they consistently observe these black lymph nodes. They increased with age, so younger donors had less. And there is a particular black particulate that's an environmental thing that they are called polycyclic aromatic hydrocarbons. And they're from environmental pollutants such as motor vehicle exhaust, heat, like smoke and wildfires. So I don't think there are a lot of wildfires in New York City, but there are a lot of vehicles and exhaust. So that's one way that people could pick these things up. When they looked, I was wrong. The majority so first of all, the particulates were in macrophages. They weren't in other cells, but they weren't in these subcapsular sinus macrophages that collect things. They're actually in the T cell zones. So they could find these macrophages in the T cell zones of lung draining lymph nodes that were chock full of these black particulates. So that's cool. Does it mean anything? It's very hard when you're taking lymph nodes out of human cadavers to do any biology with them other than characterizing phenotypes. So they kind of looked at surface markers on cells as a proxy for their function. And they did see that activation markers decreased with age in the T cell zone macrophages, but not in the mesenteric lung macrophages. So that suggests, well, maybe something's happening in the lung. And when they did some imaging, what they saw is that the macrophages that had the particulates had a much lower activation marker expression and phagocytic marker expression than the non-particulate containing macrophages from the same lymph nodes. So the idea here would be that those particulates are actually doing some things. But how do you actually address this? So they went to an in vitro system with THP1 monocytes, and they fed them these particulates and they gobbled them up, which, it's what the macrophages do. And then they incubated them with fluorescent bacteria with a pH-sensitive dye. And what they found was that the macrophages were defective in phagocytosis, and then their maturation of the phagosomes was also impaired, suggesting that that the particulates that you breathe in accumulate with age in these macrophages that are in the T cell zones, which are, in these, areas that are really important for maintaining immune functions and germinal centers and all this other stuff in the lymph node, right? And so they're impaired in their phagocytic ability to protect against infection. And they do a whole bunch of other things that I won't really go into detail into. But the other cool thing that they did was they observed that the B cell follicles in those lung lymph nodes were dispersed with age and lost their follicle integrity. And it didn't really necessarily correlate with the particulates. It just sort of was like an age observation. But what they did see was that in areas where there were particulates, the lymphatic vessels were much fewer. So there could be some communication between these defective macrophages and the maintenance of the lymph node function. So the idea here is you can't really do much about it now, but if you've been alive for a while and you breathed a lot of particulates, it's probably impacted your ability to protect against infection, which could, in hindsight, explain why older individuals might be more susceptible to certain infections, and perhaps why regionally there were higher, hand waving to COVID-19. There were some differences in the impact of the disease on people maybe living in cities versus outside. If you parsed it out, maybe it could be due to breathing a lot of exhaust.
Oh boy, I guess we should be wearing masks our whole lives. Huh?
It definitely speaks to the disparities in individuals who live in more polluted areas compared to not, and could contribute to why we see differential health effects. So it's fascinating, the cohort that she's built to be able to see these things in humans and not just animals.
Of course, it's not proving it. You just don't know, right?
Right.
I would be curious to know what percentage of lymph nodes in the chest would you have to have with these particles in them in order to impair responses? Or one probably is not going to make a difference, right? How many lymph nodes are in the chest cavity? Do you know offhand?
Nope.
A lot.
A lot? Like 20, 100 or more? More than 100?
I don't know.
Okay.
I don't know the exact number.
Maybe
Let's see.
Somebody knows who's.
I think that they are. My understanding is that they are rather difficult to
500 to 600 throughout the body
to identify upon dissection. And so I think it might be hard to have an exact count because identifying them as individual lymph nodes might be challenging.
Or maybe we could say 100. Maybe we could, if there's, like Vincent said, 500 to 600 in the body, maybe 100. I don't know.
Yeah, so you wonder what percent needs to be particulate in order to have impaired, right? That's the key. And if, I don't know, did they mention in their autopsies whether anybody had massive particulate, or was it just a few lymph nodes, right?
I don't know if they parsed that out, and I don't know if they get that much material. They probably get a few lymph nodes.
Yeah.
From each individual. And you're only going to get ones that are larger that you can see, right?
Sure.
Right. Yeah. This reminds me of something I was talking about with students recently, that, there are a lot of ideas about how your immune system works that, your grandmother told you, right? Like, breathing polluted air is bad for you and don't go out in the cold and all that kind of thing.
Yeah.
And it's actually kind of cool to realize that we're now able to use some really sophisticated techniques and actually show some of the mechanisms behind those things. That maybe your grandmother wasn't wrong. She may not have had a lot of evidence.
Here's a great book to write: is Grandma's Immunological Wives' Tales.
Yeah, exactly.
That have now been proven true.
Right, because there are lots and lots of them. There was one in the news last week about cold exposure.
Yep.
So I think it's really interesting that we're realizing some of the ways that a lot of those things, have this mechanistic backing that we couldn't necessarily show before.
So how does chicken soup work, Brianne?
That's a great question. My guess is about the salt and the hydration.
Salt, hydration, and probably
heat
or
last night on our stream, we were talking about soup because Amy has tonsillitis. So people were saying, "Oh, you should have some chicken soup." And one lady said, "It's really just the steam coming out of the soup," and that's why she likes some really spicy, hot soup to clear out.
But spicy is different because spice is capsaicin, and that absolutely has effects on the immune system.
For sure.
Channels.
We've always known that pollution is bad exacerbates respiratory diseases, right?
Yeah.
And so many large cities have improved pollution. In certain countries, like here, New York air is much better than it used to be.
Oh, yeah.
But Mexico City is still pretty bad, right?
Yeah.
And probably Beijing is horrible, right? They say on some days you can't even see. Yeah. So there, people should worry.
Yep.
It does speak to like the multiple generations of passed down stories, right? If you keep seeing a certain phenomenon over time, with if you're not in the scientific era to prove it, okay, well, we're going to pass that down generations. It's very interesting. Okay, am I next?
Yeah, you're next, Steph.
Okay. Okay, great. So this is my second paper. I picked out 2, so this is 2 out of 2. And it's called Pregnancy Enables Antibody Protection Against Intracellular Infection. It's by John J. Erickson, and it's out of Sing Sing Wei's lab at the University of Cincinnati. And this is I was rereading this. I'm like, I gotta go back and deep dive into this paper because it's so fascinating and little complicated. So let's see if I can make it through. Okay. So we discussed that antibodies can be glycosylated, and we mostly study or look at the FC portion, but the Fab region is also glycosylated. There are also glycan sites on the Fab region that could have an impact on the ability of the antibody to do its thing. And what this paper demonstrated is that only in pregnancy was a particular post-translational antibody modification, aka glycosylation modification, happened only during pregnancy, and it enabled the protection against an intracellular pathogen called Listeria monocytogenes, which is a very dangerous pathogen to pregnant women because it can cross the placenta and cause a lot of problems for the baby. And what this paper showed is that so 2 things. If you got rid of B cells, the babies were no longer protected from Listeria. If you got rid of the B cells in mom, that is, if you immunized mom preconceptually, so if you primed her with a Listeria antigen pre-pregnancy, those antibodies were not protective in the babies. However, if you primed her during pregnancy, she was protected. If you transferred antibodies into a pregnant mother that were primed in a virgin mom, they were not protected. And so what this demonstrates is that only during pregnancy did they see a change in the glycosylation of IgG that could mediate protection against Listeria monocytogenes by engaging a particular receptor, and now I'll go into that in a bit. Okay, so did that make sense, or do I need to repeat it? Because it's a little convoluted.
So there's a specific antibody that will be made against a version of the antibody that has a specific different glycosylation only during pregnancy.
Only during pregnancy, correct.
To Listeria.
Yes. And what they identified to be the first of all, they tried to figure out, well, what was the change? Mm-hmm. And they found that during pregnancy, these antibodies are deacetylated. So they have sialic acids that come off the side of the Fab region, and the sialic acid is normally acetylated. During pregnancy, there's an increase in the enzyme that takes off. So the 9O acetylation pops off only during pregnancy. And taking off the acetylation off the sialic acid of the antibody allows that sialic acid to bind to CD22. It's cyclic CD22. When that binding happens, then you can activate B cells to help produce more antibodies. Without that binding, B cells are more in this regulatory state. They produce IL-10 and they have a decreased antibody production. And so it's the interaction of the sialic acid with this CD22 that stimulates B cell production that does not happen when the animal is not pregnant. And so there's a lot of questions that brings up, what is it about pregnancy that upregulates the enzyme that deacetylates the sialic acid? Is it a particular hormone? Is it a fetal protein? And it's really an interesting way to demonstrate how glycosylation can impact so it's a non-FC glycan that's enabling the protection against this pathogen, and how, again, evolution has found a way that to change the antibody so that it can be protective against a pathogen that would terminate a pregnancy. So, very cool.
It's interesting that 9O acetylated sialic acid is the receptor for Influenza C viruses.
That's very interesting.
And so if you took off the acetyl group, you wouldn't be able to infect them. Now, it doesn't really matter because most influenza C virus infections are asymptomatic or very mild. Okay. So,
but wouldn't that be interesting if pregnancy induces the enzyme that deacetylates 9O, then are pregnant women less likely to get influenza C?
Is the enzyme systemic?
Well, that's the thing. So they particularly looked at this enzyme inside, and did they get to what cell type? It doesn't say that it's circulating. So you're right. It would have to deacetylate the lung cells or the nasal cells.
Just an interesting coincidence, right?
That is interesting. Yeah. So,
Brianne, you're next. I'm sorry. Were you done?
Go ahead, Steph.
Yes, I'm done. Yes.
Sorry.
No, you're good.
So the second paper that I picked is a paper that people who have listened to TWIV may notice some familiarity with, as we did talk about it in a TWIV episode, but I've thought about it ever since. And it's one of those.
Yeah, I remember this. This is a great paper.
It's a great paper, and I just found so much of it really exciting. So yes, this is TWIV 882. But the title of this paper is IL-1 and IL-1RA are key regulators of the inflammatory response to RNA vaccines. This paper was published in Nature Immunology in March of this year, and the first author is Tatanen, and the last author is Ira Melman. And here they start out talking about mRNA vaccines and the inflammatory responses that they seem to induce. And that's something that has been discussed in a number of settings. And they specifically note that those levels of inflammatory responses influence the doses that could be used of these vaccines when they were used in clinical trials, and that was not predicted from the preclinical mouse studies. And I recall working on some similar types of nucleic acid-based vaccines in graduate school, and at one point I remember doing some math of how much nucleic acid we needed to stimulate a response in a mouse, and we were going to extrapolate up to how much dose we would need in the human, and we came up with something absurdly large. And so I recalled that when I was thinking about this paper. And what they decided to do here was to look at some of the inflammatory responses that happen when human cells, particularly some PBMCs, were stimulated with RNA lipid nanoparticles, as are done in the mRNA vaccines. And they showed that the human cells, and they also showed this similarly from some mouse cells, made IL-1, particularly IL-1 beta. The mouse cells made a little bit more IL-1 alpha compared to IL-1 beta, but they did see some IL-1 beta in both cases. And they could see inflammasome inactivation. And they could also show that IL-1 led to cytokine release of many of the other inflammatory cytokines. And so if the IL-1 was blocked, then there was no TNF production or no IL-6 production. And so it's sort of this cytokine release syndrome was very dependent on the IL-1 beta. But when they looked more closely, they realized that they were seeing a different response in the mouse cells than in the human cells. And that was sort of hinted at when they saw the IL-1 alpha in mice, but they actually saw that the mouse cells made large amounts of IL-1RA, which is anti-inflammatory cytokine-like molecule that binds to the IL-1 receptor and turns down inflammatory responses. And mice make massive amounts of IL-1RA in response to the lipid nanoparticle and the RNA. And it does seem like both the lipid and the nucleic acid are important in this signal. And when that response was blocked, the mice actually made a better adaptive immune response to the vaccine elicited antigen. And so I just thought that thinking of that, the mice are not always equal to humans is something that's really important for us to think about. Especially, I've thought about a number of different ways that the innate immune response differs between the two. And this is very clear in this difference in innate immune responses and shows us something that I think is, really interesting and related to something that many people are seeing in their daily lives and is helping us to understand that phenomenon.
Yeah, the one thing I really loved about this finding that because of this IL-1RA in mice, you can give them 1,000-fold higher in vaccine doses, right?
Right, exactly.
Wow.
And how could we do that with us? Because it probably would be beneficial, right?
Right, it would be beneficial. And so we need to think about this IL-1 production in us. But will that influence our adaptive immune response? Yeah. Because mice are making a reduced adaptive immune response. And so it's a little bit of a trade-off. We get these side effects, but we don't have IL-1RA potentially reducing our adaptive responses.
Yeah. Very cool. Yeah, I think you were on that TWIV, right, Brianne?
I was, and the whole time I was
exciting.
A big fan of the paper, and it gave me a lot of ideas of experiments that I wanted to do. And I've recommended this paper to a number of people since that episode.
All right, I have another FC receptor, FC part of the antibody paper. This one is in Cell. Antibody effector functions are associated with protection from respiratory syncytial viruses from Galit Alter's lab, and she was also on the previous paper. And the first author is Yannick Barch. So basically, respiratory syncytial virus, as everyone knows, serious lower tract infection of the very young and the very old. We have a monoclonal for treatment, but that's about it. It's incredibly expensive. The problem is that neutralization does not correlate with protection. And we have a lot of data in people that show that when people are protected, it's not because of neutralizing antibodies. The same thing in an animal model, but there's a hint that antibody FC effector functions are important. So here what they did is they have adults who are immunized with a vaccine comprising an adenovirus vector with the pre-F protein of the pre-fusion protein of respiratory syncytial virus. And so these individuals are immunized, and then they're challenged with RSV. And you can look at protection from infection, what correlates with it, and what correlates with it is not neutralizing antibodies, but what they call opsonophagocytic functions, the ability of the FC portion of the antibody to bind to FC receptors on macrophages and be taken up. They look at, IgA in particular are important for this. Also, the glycosylation of the IgG is also important as well. And so this is cool because it's in people. They actually do some engineering in mice where they can give the mice antibodies whose FC has been improved to bind better to FC receptors, and that correlates with better protection. They call it rational FC engineering. And so another paper showing it's not neutralization of the antibody, but the FC portion that's driving protection, which I think is cool that it's more than one virus.
Yeah, just as a side note, anytime I hear human challenge trial, I was just talking with somebody about a norovirus human challenge trial, and it is still crazy to me that those happen. I mean, RSV in non-immunocompromised adults is, not generally going to cause you severe disease, but it is still amazing to me that those happen. Yeah.
So the norovirus can be done because you just have diarrhea and vomit for a couple of days, right? You don't die as long as
you don't.
But you can
it's still horrible.
It's still horrible, yeah. But you get $300, so it's a lot of. You can get people to do that.
Yeah, right.
So the RSV is an attenuated virus that was made many years ago.
Oh,
so it doesn't cause severe disease in people, so they can do that. Same thing with influenza; they can use vaccine strains and so forth. And as you know, SARS-CoV-2 challenge was done in the UK.
Right. And I was going to say now, I mean, it is interesting with SARS-CoV-2. If you look at the proportion of people in the healthy, in like the middle-aged group, they're not likely to get severe disease. Will we see more SARS-CoV-2 challenge trials? I don't know, but.
Yeah, I mean, I think the SARS-CoV-2 challenge is iffy because, look, people are always saying to me, I'm worried about getting COVID because I don't want to get long COVID. Okay. Yeah. And people of all ages are saying that. Right. So how do you know these young people who you think are so healthy, how do you know they're not going to get long COVID then?
Yeah.
Right?
Yeah, you're right. Until we know more about that, that's
yes. I think we don't know enough, right? So
I would not be signing up.
No, me neither.
But that's a cool paper, Vincent.
It is.
All right, we're back to Cindy.
So we should keep going. One more short one. Okay. Yeah. So the third paper I picked, I'm calling color-coded antibodies. I'm not going to go into a lot of detail on this because it's really, really complicated. But I will say, so this is a paper called Molecular Fate Mapping of Antibodies on Primary and Secondary Immunization. It's on bioRxiv. It is from Gabriel Victora, who we had on episode 52 earlier this year in January. I just think he just talked here a couple weeks ago, and he talked about this work, and I was like, I have to go read this. And I read it, and I'm still digesting it, but I just thought it's such a cool idea that I thought we could just snippet it. And so if you remember, he has used these confetti mice. So basically, these are mice that will randomly generate different colors and different B cells. And so you can look at development of B cell clones. And he has these beautiful pictures of lymph nodes where you've got all these hundreds of different colors, or not hundreds, but they can color code them. And then as an immune response develops, you get now one follicle turns completely one color. So basically they become monoclonal, which is really kind of cool. He's also used these dynamics to show what we've mentioned already is that, so it seems to be when you do a recall response, there are these naive B cells that are pulled in to contribute to the second immune response, so the boost or whatever. But what we can't tell, we can never, to this time, we could never distinguish between antibodies that were made, or antibodies that were being produced by B cells that were activated in a primary immunization or infection with those that could potentially be made following that. So naive B cells that come in and contribute to the production of antibodies. Because an antibody is an antibody and you can't tell them apart unless I guess it's during pregnancy where they're differentially glycosylated, right? But until now, you can't tell who's made the antibodies. So what they do is they fate mapped antibodies. So we can fate map cells, right? So you could create a mouse in which when you treat with a drug, they'll CRISPR out a stop codon and all the cells will light up a particular color that are now mapped. And then you can look at them over time and see all the different subsets of cells that develop from that cell. Well, they took that idea and now they did this with antibodies. So how did they do this? So they took B cells, and B cells have, they took the kappa light chain and they put a flag tag on the very end of the kappa light chain. And then they put a stop codon, and then they put a strep tag. But then they put LOX-P sites on the 5' of the flag and the 5' of the strep. So a B cell gets activated, it's going to make a flag-tagged antibody. If you give the mouse tamoxifen, that will activate the CRE. The CRE will cleave out the flag, and now the antibodies will be strep-tagged. So you can immunize a mouse and create a subset of B cells. Then you can treat with tamoxifen, and all those B cells, all the antibodies produced by those B cells will be strep-tagged. Then you can wait. Then you can immunize again, and any new B cell antibodies will be flag-tagged, and the old ones will be strep-tagged.
That's cool.
So you can parse out which antibodies were made when. So why is this important to do? I mean, it's a cool technique, right? This is really neat. But if you think about something that people have been talking about, he calls it primary addiction, but it's often called original antigenic sin, right? So the argument would be if you immunize with something, let's say it's original SARS spike, right? And then you come in with a different one, you primarily generate the immune response to the original if it's enough cross-reactive, right? And so they have now an ability to test this, because they can immunize with something and they can switch all the antibodies and now immunize with something that's identical, similar, or distant.
That's cool.
And they can measure the reactivity of the antibodies. So they can measure when they were made, so who they were made by. And then they can tell, are they more reactive to the original or to the new one, and who's making antibodies that are now neutralizing or whatever to the new strain and whatever. So there's a lot of data in this paper, and there's a lot of interesting stuff that they did. But the bottom line is that original antigenic sin is real. Okay, so if you inject with something and then you inject with the same thing, or you inject with something that's very, very, very similar, the primary response is driven by the original antibodies. So there's a little boost of new antibodies, and they go away quickly, and they don't really contribute to a boost.
Right.
So that's things that are really close together. But they could test this, and the further and further the antigenic distance was, so the less similar the initial immunizing antigen from the boosting antigen was, the less imprinting or primary addiction they saw. So more new flag antibodies are made to the new thing that you inject. So the more different the two antigens are, the more new antibodies you make. The closer they are, the more original antibodies are made in preference to the new one. And so why do we care? We all care because we want to know about SARS-CoV-2, right? So they did do this, and they found that if they primed with Wuhan, a spike, and then they either boosted with Omicron or Wuhan, the boosting, they were able to boost basically to both. So you didn't have that. There was a preference for the primary antibodies, but you generated a good response to the Omicron, even if you primed with Wuhan, and that this was boostable. And they were with the new antibodies. So that was all good news. But it doesn't really address the way we are getting boosters, which is mixing them together.
Yeah, right.
They didn't test that because that could influence how things are playing out.
I was going to ask you about that. Yeah, because when you get this bivalent booster, if you had the original, you make pretty low antibodies to Omicron, right?
Right.
And so why is that? David Hoth says it's original antigenic sin, but I'm not convinced that's the explanation, right? If you just gave Omicron alone without the other, right, which you don't do, what would you see? I
think that in mouse, what it suggests is you are able to generate antibodies to the Omicron and overcome this imprinting because they're far enough different.
Yeah. I think that's the point. And that speaks to the issue with flu, right? In the same problem of original antigenic sin, or what did you say he calls it?
Antigenic imprinting.
Primary addiction.
Primary addiction or antigenic imprinting.
We have 3 different names depending on your religion, I think.
That's awesome. Yeah. Yes. And so it would be interesting to do this with different strains of influenza, ones that are more or less similar, to try to piece out similar questions.
So let me understand what's going on here is that you've immunized someone, and then you have B cells with receptors. And so whatever you give them next, if it cross-reacts with those receptors on the B cells, it's going to give you the original antigenic, because those cells are going to make the antibody that's cross-reacting, right?
Right.
A portion of them.
Right. So if you put the new antigen in, if it's close enough to the old one that it cross-reacts, it's going to drive expansion of those old ones, which are not optimized for the new antigen.
And will that interfere with making new B cells?
He's saying yes, because they are seeing a blip in new antibodies that goes away quickly. And when you boost, it looks not so well boosted.
I almost picked a paper about that too.
This is very cool. This is great.
You still would have memory B cells to the new antigen that need additional time.
Right.
They just are going to be lower.
Because they boosted again, and it still wasn't. So if you boost, I don't know which direction I'm going because it's always come backwards. If you boost, it should go up, come down, and then when you boost, it should go up higher and come down, right? It didn't. So if it was close enough in distance to the thing that they injected originally, the new antibodies that came up, the flag ones, would go up and down, and then if you boost it again, they would go up and down, almost like a primary response. There was very little boosting that happened.
What you're saying. I understand.
It was all focused on the original.
But
the further you got apart, the more you'd get the typical boosting that you would want.
Which might be okay, because is it cross-reactive enough? Are you still going to have your T cells working for you, your non-neutralizing antibodies working for you? I guess that's the question of, is it enough for the new thing?
Right. I don't know. But it's interesting that they can now tease it out, and they can look at, if it's 99% similar or 90% similar or 70% similar, like you can now start to look at this. And they did do flu in this as well. I'm not going to go into all the details, but they did like challenge with flu and then challenge with another flu strain that was a little different and further different. And they show kind of the same thing. So if the flu was quite similar, the primary, the boosting response was driven against the original antigen and very little against the newer antigen.
Right. Isn't it so funny that I just think about like what viruses are thinking. We spend so much time and effort trying to figure them out and like be a step ahead of them, but they're always a step ahead of us. It's this constant battle between.
Well, they have a huge head start, right?
Millions of years,
And they've accumulated all this evolution and information, and we're just chipping away at it. It's going to take us a long time. I mean, it's already there. It's not like they're developing new things, right? So Steph, you're done because you picked two.
I did too. Do you want me to do an email now? I can hop over to the emails.
Let's finish the...
Sure.
We have two papers left. Brianne is next.
Yeah. So this paper is a little older than some of the ones everyone else had picked, but it is part of a talk that I heard this summer. And I thought the talk was really fascinating. And so I wanted to mention this paper. So this paper is called Endogenous Retroviruses Promote Homeostatic and Inflammatory Responses to the Microbiota. It was published in Cell in July 2021. And the first author is Lima Jr. The senior author is Yasmin Belkade. This was it's a pretty complicated paper as well. And I will say, to me, it was a nice thing to see in talk form and get to hear the rationale and how it tied into some other work and things of that nature. They started out by looking at mice that were colonized on the skin with Staphylococcus epidermidis, which is a commensal organism. And they saw that these mice were making skin immune responses with a number of different T cells, as well as type 1 interferon. And they don't really talk about it as much in this paper, but they sort of have this idea that ideally it might be not great to be starting with an immune response and an inflammatory response of absolutely zero, because those viruses have that head start, as Steph was just mentioning. Perhaps we need to have a little bit of sort of background inflammation, a little bit of background interferon, so that when a virus comes in the first place, we are ready to protect against it. So a lot of what they're thinking about here is kind of what is this level of sort of appropriate background? You don't want to have too much, you don't want to have too little. But they also kind of think about exactly what's going on with the transcripts when these mice are colonized with the commensal organism. And they note that they actually see a lot of transcription of endogenous retroviruses when they have the commensals on the microbe. They see quite a bit of endogenous retroviruses. And if they block that, they don't see any immune responses. So it seems as though the endogenous retroviruses are actually, the mice are transcribing ERVs, and that's inducing the skin immune response. If they block reverse transcriptase, no interferon, no skin immune response, any way of blocking those ERVs. And so they sort of come up with this model of the commensals are inducing transcription of endogenous retroviruses. That's turning on DNA sensing, and that's sort of giving you this background level of inflammatory response, which was all really exciting. But then they finished the paper by showing that there are other things that could turn on those endogenous retroviruses. And so the commensals are one thing that are sort of influencing your background level of inflammation. But then they also show that if they change the mouse's diet and give the mouse a high-fat diet, that turns on the endogenous retroviruses. And so then perhaps the mice, as they give them too much of a high-fat diet, have a really high inflammatory set point, and are more prone to inflammatory disease because they've kind of pushed this set point a bit too high. And so, again, there were a number of papers and a number of studies talked about in this talk, but I found that talk really fascinating. And I went back as soon as I heard it and read this paper that was covered throughout this talk.
I love that concept, right, of immune set points.
Exactly. That was one thing that I really took away from it.
That's very cool.
Steph, you're moving away from your mic.
Sorry, I was trying to stretch my legs.
Oh, okay.
Sorry, I was getting cramped up.
Go ahead. You can stand up.
It's okay, I'm good.
It's interesting. Go ahead.
No.
I was just to say, Brianne, it's really interesting because I've thought for many, many, many years, I've been working on nucleic acid sensing TLRs and thinking, really, what is the strong evidence that they are involved in immunoprotection? Right. It's not great. You can find an isolated virus or bacteria where if you knock it out, now you have susceptibility. But with other TLRs, it's a bit more clear. And so I've always pondered, and many people now talk about this, whether those nucleic acid sensing TLRs are playing a normal role in host.
Wow. Yeah.
I think it might
evidence of that.
partly be in wound healing. Right. Or in non-infectious inflammation. And then I guess also potentially involved in setting this set point.
Yeah, exactly. And so I just thought of the idea of the set point, as well as the idea of, maybe even going back to Cindy's black macrophages, outside things influencing what kind of set point you're having here, all in the case of high-fat diet, and how that could really change levels of immunopathology that we see in response to any particular thing. So It's an older paper, but I only learned about it this year. It was new to me, and I got a lot out of it.
It's an older code, but it checks out.
Yeah.
It would be nice if you could remove all these ERVs, but you can't. There are too many of them.
Well, it might be a problem if you do. Maybe they're also playing an important role through this, keeping our set point up.
If you could remove them in mice, just experimentally to see.
I see. I was like, I don't know if we'd want to remove them all.
Not from us, but
I'd
like to. But however, Cedric Fichat is doing this in zebrafish, where it's
so cool.
It's easier to do because there are fewer. But of course, there we don't have the. Do you have a similar immune system? I guess.
Yeah,
I don't know.
some. I mean, they definitely have B cells and T cells.
So maybe you could see an effect on immune development, right?
Yeah, all they could do here was look at treatment with reverse transcription to sort of try to reduce what's going on. But it would be really cool to see what happened if you didn't have any.
The pigs are used for transplantation, so people want to remove the PERVs, porcine endogenous retrovirus. And so George Church apparently has removed many of them from pigs, but they don't. They're sickly, as you might guess.
Huh.
It's interesting. Okay, my last one is about a similar idea that we're talking about, that neutralizing antibodies are not the whole story. This is a paper in Science Immunology. CD8 T cells contribute to vaccine protection against SARS-CoV-2 in macaques. It's from Dan Baruch's lab. The first author is Jinyan Liu. And I'm only just going to read you the first couple of sentences because it's perfect. Neutralizing antibodies are generally considered key correlates of vaccine protection against SARS-CoV-2. Recently, robust vaccine protection of severe disease with SARS-CoV-2 variants that largely escape neutralizing antibody responses has been reported, suggesting a role for other immune parameters for control. However, data demonstrating a role for CD8 cells in vaccine protection haven't been reported. So that's what they do in this paper. So, that one sentence, we still get robust protection against severe disease. So why we have a bivalent booster again? Why, if the thing is still working? Oh, that's Paul Offit. Never mind.
Yeah, go check out TWIV something.
They vaccinate macaques with an adenovectored vaccine, then they challenge them with Delta. So the original is the original Wuhan spike, and then they're challenging with Delta. Giving the animals have good control of virus, and if they give them antibodies to deplete the CD8 cells before challenge, they get higher levels of virus in the upper and lower tract. And so they're saying this, these CD8 T cells are contributing to control of viral loads, which makes sense, right? I guess they're eliminating virus-infected cells. So I think it's cool.
It's a good reminder that your immune system doesn't want to only rely on one thing, because the virus is going to mutate, and if those neutralizing antibodies don't work, so you better have redundancy.
But let me ask you, if you could take serum and you measure neutralizing antibodies, if you take out lymphocytes and measure T cells, what does it tell you, though? That's the problem, right? It's not demonstrating control of infection just by saying, there are virus-specific T cells here, right?
I think you could assume, right? You could assume if you have greater function of those cells, that would correlate to protection. So, but you're right. It's less of it's more challenging to link that.
You'd have to do a lot of much more complicated assays on those T cells to really show their function. I guess you would have
to do, in the old days, they used to do cytotoxic assays, right? They used to do chromium release. Right. Remember that?
I do. I have many nightmares about chromium release assays. But what you can also do things like resimulating the cells in vitro, and then looking at cytokine production or degranulation or other types of things to get at that. There are also some non-radioactive cytotoxicity assays one can do as well.
Mm-hmm. But it does go back to, when you're designing a clinical trial, it isn't the case that those assays are done as frequently because they're not as easy.
Yeah.
Correct.
So, it's almost like immunity isn't just one thing.
I would agree. Yeah.
Well, that's the theme, right? All right. So we got through that. We have a little time for letters. And cool. Steph, you're excited to do a letter. So why don't you take that first one?
Oh, boy. Okay. Let me go. Okay. Nicholas. That's who I'm on.
Yep.
Dear Immune Host, I thoroughly enjoyed the discussion about the Cell Host and Micro paper regarding improved wound healing through IL-1B signaling. That's episode 57. I have the feeling that there is a shift happening in the way inflammation is discussed. While in the last years, the focus seemed to have been on detrimental effects of unregulated inflammation, cancer, cytokine storm. Now I read more and more research highlighting the positive role of inflammation, for example, in tissue remodeling and regeneration. But this just might be my limited overview of the field. I would be interested if and how your views on inflammation have changed over the years. The discussion about possibly using microbes to facilitate wound healing reminded me of a cream I often got for my wounds when I was a child. I remember reading the label and back then being very surprised by this cream with the brand name Pyolysin contained lysate of all kinds of bacteria, including Staph aureus. Thinking about it later, I assumed that the microlysate might contain antibiotically active compounds secreted by the bacteria, but now I think it could be interesting to find out if it induced IL-1 beta dependent wound healing through TLR ligands contained in the microlysate. Maybe by luck, the company that makes this cream since the 50s was tapping in some really sophisticated activation of the self-healing properties of our skin. Thanks for your podcast. And special thanks to Vincent for his virology course, which right now helps me feel better prepared for my PhD at the Institute of Virology. I'd have to look up pyolysin. I don't know anything about it.
I am Googling it, and I'm not really seeing anything.
You guys, have your views on inflammation changed over the years?
Yes, it has. I think that it is it's a state, and it is context dependent on if it's a viral infection, and it has to be self-limiting, right? It can't be forever. So I don't know if that's changed, but I mean, I'm learning new things every day. So like, I don't know. To me, nothing is static. I'm just constantly learning.
Yeah, I think that I have thought about a lot of parts of the immune system in a very Goldilocks fashion for many years. Mm-hmm. And so I think that to me, inflammation has always been one of those things where there is a positive role for it. Maybe we don't talk about it as much. And I think that, maybe we are now starting to talk about the positive role, but there's definitely a positive role for inflammation. There's just always a negative side too. It's you don't want too much, you don't want too little. And I come back to that idea of Goldilocks more and more often in so many parts of my understanding of immunology that I guess we're just talking about it more for inflammation.
Yeah, what you're saying about the Goldilocks zone, I think I definitely agree with that. I've always been thinking that inflammation is important and required because anytime you have damage, you need to repair it. In order to repair it, you need to have some sort of inflammatory response that will drive the production of the mediators and things that are necessary to repair and return to homeostasis, right? So I kind of think about any kind of disruption, whether it be an autoimmune or an infection or a traumatic injury, is driving you out of homeostasis. And the inflammation is necessary to get you back to homeostasis. And you could go too far or not be able to reach that and be chronically inflamed. But the job of it's really just saying there's an immune response. Whether that be innate or adaptive, you have an immune response to try and bring you back to homeostasis. In the case of the innate part, that's basically going to involve inflammation of some kind or another to drive it back towards homeostasis. So I've kind of always thought about it as important.
Yeah, I think that one thing that ties into what Sydney says is that I haven't always thought about the repair part as being as related to immunology. And that's something that I've been moving towards thinking about more. So I appreciate what you said about, tying to repair, because the repair piece and the importance of repair is something that is a bit of a change in my thinking.
Yeah, I guess I've always thought of the immune system's job is to keep you in homeostasis. And so something that drives you off, you got to bring it back. Go off this way, you bring it back. And so I always thought of inflammation as being important.
Well, you can still buy pyolysin.
You can.
Oh, is that true?
Yeah.
Huh, interesting.
Yeah.
Now I want to do some experiments with it.
You could get it. Yeah, do some, see what it's going on. I bet it's stimulating inflammation.
TLRs.
Yeah.
TLRs,
Nucleic acid sensing TLRs to repair the tissue.
Yeah.
Cindy, can you take the next one?
Sure, because we met Marguerite. So
Yes.
Marguerite writes, she actually took care of my sons. She was a pediatrician. You'll hear this in a second.
Cool.
Dear immune podcasters, first I want to say that I've really appreciated your discussions of immunologic topics over the past 2.5 years. I practiced pediatrics in Ithaca, New York from 1971 to 2019. Wow. So she did see my children when they were sick. When the pandemic started, I found TWIV and the other podcasts. They made both retirement and the lockdown a new learning experience. Special thanks to Vincent for the course in virology and to Brianne for the course in immunology, and to all of you for the education and updates on SARS-CoV-2. in the June 23rd, 2022 Nature article, I read targeting SLC7A11 improves efferocytosis by dendritic cells and wound healing in diabetes. Then I listened to the podcast, Bacteria, the New Cure for Wounds. That was the one we were doing. We talked about it already. She says, I was not familiar with efferocytosis, so I followed up by reading some review articles. And sidebar, for those who don't know what that is, that's when a cell is dying, macrophages will come in and find them and eat them and clear them up. So that's efferocytosis. So returning to her note, with cells turning over all the time, it makes sense that if apoptotic cells are not efficiently dismantled, they will impede or otherwise cause problems, especially by allowing necrosis to proceed, which could release molecules that could lead to autoimmunity and persistent inflammation. That the skin microbiome affects wound healing does not surprise me. A favorite surgical prof in medical school used to say, dressings are for salads. And a skilled plastic surgeon with whom I shared patients favored soap and water to manage burns. But it was very nice to see a signaling mechanism that could at least in part explain the microbial contribution. It occurred to me that perhaps the skin microbiome produces molecules that could affect the cascade of reactions that put the brake on the membrane transporter, SLC7A11, and promote more efficient efferocytosis and better wound healing. The germs may be doing a lot of things. Efferocytosis seems like a very important area of cell biology that is sort of under the radar. I would appreciate any words of wisdom you can provide. Many thanks for your excellent and much appreciated podcasts. I look forward to each one. Marguerite Uphoff.
Yes. I mean, we could do an episode on efferocytosis. It's definitely not my area of expertise. So
I could pick a paper on that. I think it's really cool. Interesting. When I teach it, I always get giggles. I'll see. Do you do this too, Brianne? When you teach this, there are find me signals. So dying cells release ATP, which macrophages can sniff and follow along to find the dying cells. And then there are eat me signals. And so whenever I say there are eat me signals, the students laugh and giggle. But yeah, so when a cell is dying, it will flip. It has an inability to maintain the integrity of the plasma membrane. And when it does that, lipids that are normally stored facing the inside of the cytoplasm flip to the outside. So this is phosphatidylserine is a common example. And that is a signal to macrophages that that's a dying cell and they should eat them. And the cool thing is that when a macrophage eats an apoptotic dying cell through this efferocytic process, it changes the functionality of the macrophage and they become more immunosuppressive and quiescent because they're eating things that just need to be cleaned up, right? It's not eating things that are inflammatory and dangerous anymore. So there, again, this is idea of them being involved in this wound healing where sometimes you want to generate the inflammation and sometimes you want to shut it down. So when there's no more danger around anymore, you want to be quiet and clean things up. I kind of call those macrophages the janitors. They come in and they clean up all the debris, and they don't need to call the alarm anymore because they're coming in at the end of the inflammatory reaction.
Yeah, and if anyone has ever done an apoptosis assay using annexin 5, you have taken advantage of that phosphatidylserine flipping that Cindy mentions.
Yep. And you can visualize this. So you can label dying cells and feed them to macrophages, and you can see them inside the macrophage when they gobble them up. It's
so cool. Efferocytosis is involved in tattoo maintenance, right?
Yeah, probably, because when the macrophages that have the dye in them start to die, they're going to apoptose, and then the other macrophages are going to come and eat them, and then they're going to keep that dye. Yep.
So next time you see somebody with a tattoo, you say, hey, did you know those depend on efferocytosis?
Did you know your macrophages are eating your tattoo dye?
They're like, this is why we don't invite you to parties.
Exactly.
I think that's a great T-shirt in some way.
Yeah.
Okay. Brianne, can you take Rona?
Sure. Rona writes, Dear Immune, my favorite of the This Week in sister shows. I have follow-up questions to the questions that were asked at the end of episode 57. Topic 1, as a vegan who exclusively breastfed her young, who is not squeamish about breastfeeding, but is squeamish about consuming other animals' milk, what immune benefits would happen, in theory, if people continued to consume breast milk into teenagehood? I'm going to leave to ask Steph about that one.
Yeah. So the majority of the components in milk that are bioactive, I think, would have a difficult time surviving in the stomach and intestine of an adult who has many more gastric enzymes. Right, gastric enzymes and proteolytic proteins, all these things are higher in adults than infants, which milk has evolved to sustain itself in. But let's say, a couple things I think of. So breast milk is chock full of not just the antibodies, which I talk about a lot, but a lot of innate immune proteins, enzymes, nutritional components that can inhibit pathogenic viruses and bacteria. So I remember reading in the oral cavity, SARS-CoV-2 can replicate in your mouth. So you could reduce the amount of that with breast milk containing some of these factors. And if it did survive in your gut, I don't see why it couldn't also protect. I think there are ancient literature even. I was thinking I think Chinese were old. So like the soldiers would drink colostrum or breast milk from lactating mothers after they were injured on the battlefield or something. I'd have to go back. But so it's possible that it would be beneficial, but I think the babies need it more, right? Because their immune systems are much less developed. So, yeah.
All right. So topic 2, for women who have had half a dozen lymph nodes removed only the left armpit because of breast cancer, how would receiving vaccines on the right side for the rest of her life differ immunologically from receiving vaccines in either arm? So I'm not sure if having those lymph nodes in the left armpit removed would actually be all of the lymph nodes there.
Probably not.
There might be some remaining. But I think that whether the lymph nodes were removed or not, if one always received their vaccines on one side, perhaps with, say, a annual flu vaccine, my recollection was from our episode with Gabriel and Victoria, that indicated that maybe you would stimulate the same lymph node in the same germinal centers over and over again. I, for very silly reasons, pretty much get all of my vaccines on the left side. And I remember after that episode being very proud of myself that perhaps my reasons were much better than I had originally expected.
Yeah. I think most people will do it practically on one side just because they're right-handed and they just I don't want a sore right arm because I need to use my arm. Mm-hmm. So, but yeah, I guess if I had a bunch of lymph nodes removed from one arm, I would probably just preferentially get my vaccines in the other arm.
I would too.
Where I have more lymph nodes, just because it might be better. Who knows?
So if you get your first shot in the left arm, should you get everything else in the left arm as well?
That's what we're thinking. Because we just had a paper about these germinal centers lasting for a really long time, right?
Right.
So it would be basically another stimulation of those ongoing germinal centers. So that's probably a good thing.
Because if you did it in the other arm, then the likelihood that vaccine is going to reach the other lymph nodes is low, right?
Mm-hmm. Yeah, I mean, they might both reach the spleen, but they're probably not both going to reach the same axillary lymph node or something.
If you get it in the right arm, then you're going to make new germinal centers, which aren't going to be as educated as the other ones then, right?
So should we get our bivalent booster in the other arm?
Oh, because you want to try to generate those new memory B cells, right? That's a good point. Oh,
but I don't want my right arm to hurt.
I wonder in the trial if they actually looked at that or not, right?
I think some of that, and I'd have to dig back into all of the details, is in that paper that I talked about with the fate mapping antibodies, because they did do immunizations in different spots.
Yeah.
There was one in the paper that you did too, Brianne, because the pump experiment, I sort of remember them, didn't they do different sides? There was an experiment where they looked at whether it was on the same side or the other side. I need to go back because it's been a little while since I read that. And I don't remember the details of that. I don't want to get it wrong, but I'm pretty sure that in that paper they did something with doing on one side and then the other side, and there was something to learn from that, and I don't remember what that was. We'll have to go back and look at that.
Well, Rona finishes off. Thanks, awesome scientist.
Oh. All right,
that's cool.
I think. Oh, there's one more. There's one more which is appropriate for me because it's just praise. It has no science. So I can do that.
You're like, sure, I'll do that.
And then we'll stop because it's pretty near the end. As John writes, I decided at 63 to focus on health and as such devour PubMed and medical podcasts. I've recently started listening to Immune and can honestly state that I have absolutely no idea what I'm listening to. CD4, CD8, Treg. I know a little of the other 99.99% is like listening to a podcast in Sanskrit. However, the way the indecipherable material is presented is beautiful. Absolutely no pretense to engage the public. Fantastic guests. Even the way you guys talk make compelling listening. I just don't know if this is a compliment or not.
I don't know.
I think it's saying that we do a really good job, but we are targeting an audience that's immunologically educated or at least has a little bit of background.
He likes our passion.
There you go.
Yeah, no pretense to engage the public is... I don't know.
We're kind of trying.
We try.
Trying. Yeah.
I know we get in the weeds, right?
We do.
Anyway, John continues, your immunology podcasts are way better than cardiology, nutrition, lipids, or cancer. Well, thank you. That's a
big
compliment.
Better than cancer.
I normally listen.
Great tagline.
I normally listen to podcasts to go to sleep, drowns out tinnitus, but I got excited about swapping CD4, CD8 and had to find out what happened to the flip-flop mouse. Sincerely, John. Now, I think John understands.
Putting him to sleep.
John understands more than he is telling us because he says flip. You got
it. Yep, you got it.
So thank you, John. Appreciate it. And the rest of the emails are actually suggestions for papers and topics. So we'll get to those at some point, but I want to stop now because I have another pod coming up and want to get ready for that. So that was fun, right?
Yeah,
that was great.
I like doing it that way.
Because we can chat more, and chatting is where the nuggets come from, right?
Right.
Yeah.
So
I need to go back and read some of the papers you all mentioned in great detail now.
Yeah.
Yes.
So that is episode 63. That'll wrap it up for 2022. If you have any questions or comments, we'd love to get them and read them as we did today. You can send them to immune@microbe.tv. The show notes are at microbe.tv/immune. And if you enjoy our work, the parent company is Microbe TV. We're a nonprofit and we exist on your donation. So we'd love to have your financial support. There are many ways you can do it, and you can find them all at microbe.tv/contribute. And your donations are federal U.S. tax deductible because we are a nonprofit corporation. And by the way, people always ask me, who owns Microbe TV? Nobody. I don't own it. I founded it, but when I made it a nonprofit, it went away from me, okay? Nobody owns a nonprofit, so you can't sell a nonprofit corporation.
Oh, interesting.
It has no value, right? That's the way it is. And that's pure knowledge. That's what we're all about. Cindy Leifer is at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langle, Case Western Reserve University. Stephanie Langle on Twitter. Thanks, Steph.
Thank you very much.
And Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to have a conversation about all these papers.
Hope you guys have a great holiday. We'll see you till next year.
Yeah. See you next year.
Holidays.
Happy New Year.
Happy New Year.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next year.